I'd like to introduce our speaker for today, Dr. Casey Bryce from the University of Bristol. Casey did her undergraduate work here at Glasgow Uni, where Karen and I are, which is sort of a nice poetic way to end the term with a Glaswegian giving us the last talk of the semester. And she did her PhD at the University of Edinburgh and a postdoc, and she was a junior research group leader for a time at the University of Tübingen. And I guess it's probably easiest just to summarize her work by saying that all things microbial iron oxidation seem to be within her purview, including microbe-mineral interactions, water quality, and the evolution of microbial iron cycling. So I'm really excited that she's here to give us our last talk of the first term. And with that, I'll turn it over to you, Casey. Thank you very much.
Thanks, Sean.
Sorry to interrupt. Just a quick note to remind people we are recording these. So if you'd like to keep your cameras and microphones off, that would be fine. Please also save any questions unless they're urgently points of clarification for the end of the talk. And you're welcome to throw them into the chat, and I'll be sure to look for them. Okay, sorry, Casey. Thanks very much.
No problem at all. Yeah, so thanks everyone for coming, and also to John and Karen for inviting me. It's been really nice to actually kind of put a few things together and think about one of my favorite topics now. We're at the end of I guess a bit of a crazy term for everyone. So what I want to talk about today is, as John mentioned, I have a bit of an enthusiasm for microbial iron cycling and microbial iron oxidation in particular. And I just want to go through a bit of an overview of why I find this topic so fascinating, what these microbes do, how they behave, and how they contribute to biogeochemical cycles in the past and also in a lot of present environments. So one of the things that I really find fascinating about the iron cycle is that on the face of it, it's really, really simple. So there's only two environmentally relevant valence states of iron, so reduced Fe2+ and oxidized Fe3+. And all you have to do to transfer between them is to transfer one electron. And it's kind of fascinating to me that something that is fundamentally so simple can have such a big impact in so many different areas. And one of the things that makes iron so kind of special and have a sort of unique role in the environment is, on the one hand, its abundance. It's a very abundant element, but also its solubility under different redox states. So most of the environments that I think about are approximately circumneutral pH. And in its Fe2+ reduced form, iron is very soluble in a neutral pH system. But as soon as you oxidize that iron, it becomes very insoluble, and then you form a lot of poorly soluble minerals. And this transformation and dissolution of minerals through these redox transformations control a lot of important biogeochemical reactions. So we heard from Sophie Nixon in the beginning of the seminar series about the whole diversity of microbes that can catalyze the iron reduction step. So we have mostly a lot of microbes who use organic carbon or hydrogen as an electron donor to reduce iron 3 to iron 2. And more recently, we've also started to appreciate that they can even couple things like the oxidation of methane to reduction of iron. And actually, it's nicely sort of bookended this whole series to have Sophie start talking about iron reduction and that I get to finish talking a bit about iron oxidation. So I'm glad we've kind of completed the Ferris wheel in this seminar series. So on the oxidative side, we've got three different types of iron oxidizing microbes. Probably the most well-known, the kind of most famous ones are those that couple the oxidation of iron 2 to the use of oxygen as an electron acceptor. And the use of oxygen as an electron acceptor is quite easy in low pH environments where that abiotic reaction is a bit slower. But at circumneutral pH, we have to have specially adapted microbes that we call microaerophilic iron oxidizers that kind of eke out a living in redox boundaries where there's just enough oxygen for them to compete with the abiotic reaction. So we're talking kind of like 10, 20 micromolar concentrations of oxygen where they live. And I noticed on your list for next year that you have Clara Chan coming up. And I'm glad I didn't focus on microhydrophilic iron oxidation in this chat, because Clara is the real authority on that. So I'm really looking forward to seeing more of that next semester. But the other thing that's really interesting about microbial iron oxidation, and that is maybe less well appreciated, is that you can oxidize iron 2 in completely anoxic environments. And one of the ways that you can do that is by coupling the oxidation of iron 2 to the reduction of nitrate. So we have what we call nitrate-dependent iron oxidizers that can catalyze that reaction and mean that we form iron minerals in situations where we don't have any oxygen present at all. So it's not the kind of place we'd usually think of, forming rust, but this is a reaction that these microbes can help to catalyze that wouldn't happen otherwise abiotically. And then we have another type of anaerobic iron oxidizers, and those are the photoferrotrophs. So these are a little bit different from the other metabolisms that I'm talking about, because they don't necessarily get energy from this reaction. They get their energy from sunlight, but they use the oxidation of iron, so the gain of electrons that they can get from oxidizing iron, in order to fix CO2 into biomass. So this is something which I find really interesting, the fact that you can have iron oxidation, which is both an aerobic and an anaerobic process. And that means that you can have iron 2 oxidation in anoxic and in oxic systems. And the iron cycle kind of brings together a lot of different biogeochemical cycles. And I think that's one of the reasons it's such a, it's a huge field. We are not a niche group of iron enthusiasts, where there's a lot of us. And the thing that really drives that interest is how these iron transformations link to a whole host of other biogeochemical cycles. So through the metabolic and respiratory processes, we already link to the carbon cycle strongly, and also to the nitrogen cycle. But the formation and the dissolution of minerals also impacts whether or not loads of other elements are mobile. So for example, we can have carbon, which is trapped in maybe iron co-precipitates or sorbed to iron oxyhydroxides. They have a big control on the mobility of phosphorus in the environment. And there's a huge field of people who look at the interaction between iron oxyhydroxides and metals, because their mobility in the environment is very much dependent on their interaction with iron oxyhydroxides. So these are the kind of environments that one might think about when you think about microbial iron oxidation. So I'm sure anyone who's been wandering around in the forests or come across little red streams have thought, I wonder what this is. Why is there suddenly a huge bit of, a huge red river in this small ditch? And these are very sort of typical environments where we find a lot of these microbial iron cycling processes. So the one on the left is just a ditch in Sweden, and you can see quite characteristic kind of iron flocks. And then the one on the right is a spring in Switzerland. So the spring comes straight out of a mountain, and then we have a lot of iron oxidation here. And some people might recognize this picture because it's actually the same spring that's on the cover of the Ehrlich geomicrobiology textbook. So if you're a kind of hardcore geomicrobiology nerd, you get a bit starstruck at this little stream that comes out of the mountains in Switzerland. But one thing that's really, or what I found really interesting in my own research about iron cycling, is that it's not limited to these kind of iron-rich environments. So it's not a process that you only really find in, acid mine drainage or these really irony looking ditches. It's something that's important in pretty much any system that you can think of. So I had my introduction to working on the iron cycling by working on freshwater marine systems when I did a postdoc with Andreas Kapler in Tübingen. And what was really interesting there, we were a big group on quite a big grant, and we spent a lot of time at this field site. This is in Aarhus Bay in Denmark. And we looked a lot at the distribution of different iron cycling metabolisms, and what they did, how they impacted other cycles. And what really came out of this work is that even though you have a system where maybe in the pore water you only have 20 odd micromoles of iron 2, you have a very dynamic and a very diverse and a very active microbial iron cycling community. So even in the most bog standard bit of mud that you can imagine, you have all of these really interesting microbial processes that are happening. Talking of bogs, we also find them in peatlands. So this is an area where my work has sort of taken me recently. One of my students, Monique Patzner, had a really nice paper about this that came out last week. So we've been looking a bit at iron cycling in peatlands in the Arctic, and we also now do a bit of work in Wales. And what we're starting to see is that the microbial dissolution of iron minerals has quite a big knock-on effect on the mobility of carbon and the fate of carbon in these kind of systems, which are extremely carbon rich. This is not a place we would normally think of as being a very classical iron cycling environment. But when we go there and look at iron cycling, which is essentially what I do in all environments, I go everywhere looking at iron cycling. This is what, we start to see that even though we might not expect iron to have a very dominant role in the system or in such organic rich systems, in fact, it really does. Another example of places that we see a big influence of microbial iron cycling is in soils. These are some pictures from a trip that we had last year. We went to Chile, and we look a bit at soil formation at under different climate regimes in Chile. And what we see from this work, and also from a huge amount of literature, is that microbial iron cycling processes have a really important role in determining nutrient availability within soils, within weathered soils. But we're also starting to see that a lot of these iron oxidation and iron reduction processes have an important role in turning rock into soil. So there's a picture of me looking kind of excited about having this drill core beside one of my PhD students and another one of the students from the project. And what we've been looking at down there is whether iron oxidizing bacteria can actually catalyze the first step of turning bedrock into soil. So starting off, kickstarting iron oxidation, kickstarting weathering, and then eventually leading to soil formation. So all of that is basically to tell you that iron is important. We find it's got a huge amount of diversity. We find iron metabolizing microbes everywhere, and they have a big role to play in lots of different environmental processes. But one thing that's really important when we go to any natural environment and study certain microbial processes is that everything that we study there, their whole diversity that we study there, is all underpinned by their basic physiology. And that's something that is also a big part of how I think about iron cycling, is that we need to understand what these microbes do on their own before we can understand really how they behave in really complex systems like soils or bogs or sediments. So in the rest of my talk, I want to focus a bit on the anaerobic iron oxidizers, and which actually turns out, like I said, to be quite neat, because that means Clara can talk about microhydrophilic iron oxidation next time. So I want to focus a bit on photoferrotrophs and nitrate-dependent iron oxidizers, and importantly, the interactions that these microbes have between each other in the environment. So take a little step back and kind of reintroduce the photoferrotrophs. So these are one of the types of anaerobic iron oxidizing bacteria. We kind of affectionately call them photoferrotrophs. Their kind of proper name would be anoxygenic phototrophic iron oxidizing bacteria, because they're anoxygenic in the sense that they get energy from sunlight, but they do photosynthesis without producing any oxygen. So that's in contrast to cyanobacteria, which do oxygenic photosynthesis and release a lot of oxygen in the process. And like I said earlier, these iron oxidizers don't get energy from doing iron oxidation, but they actually use the oxidation of iron 2 to get reducing equivalents in order to drive CO2 fixation. So the outcome is still oxidized iron, but it's not an energy process. It's a way of making biomass. And there's a lot of variety within these anoxygenic phototrophs. So we generally split them into 2 different colors. And this is actually a little cartoon by one of my students, Verena, who there's a lot of cartoons of photoferrotrophs from her that are coming up. She got one of those little drawing pad things, and ever since there's been lots of anoxygenic phototroph cartoons, which are great for this. So they come in different colors. You have purple, either sulfur or non-sulfur bacteria that form one group. And some examples of those might be, we have some Rhodopseudomonas strains, Rhodobacter, Rhodovulum strains that are all phototrophic iron oxidizers. And then we also have the green sulfur bacteria. And interestingly, all of the known iron oxidizing green sulfur bacteria come from the chlorobe. That's not to say that it will be the only one. So we actually only have 4 isolates of green sulfur bacteria which can do iron oxidation, but they all belong to this one group. And there's also a lot of metabolic flexibility within the anoxygenic phototrophs. So that's one of the reasons that sometimes we shy away a little bit from using the term photoferrotroph, because if an anoxygenic phototroph is doing iron oxidation, so if it's doing this, then it's a photoferrotroph. But as soon as it starts using one of these other sources, either of electrons or also as a carbon source, it becomes something else. So I think sometimes photoferrotrophy is a bit of a buzzword, and it maybe doesn't capture the full diversity or the full metabolic flexibility that these strains have. And just to illustrate that, this is a principal component analysis of some work we did with a Rhodopseudomonas palustris Taiwan a couple of years ago now. We were interested in how, in order to use different substrates, how much does one of these anoxygenic phototrophs have to change its metabolism? So what you see here is to simplify, a comparison of the similarity in the overall proteome between cells that were grown under different substrate conditions. And this is arises from analyzing about 1,500 proteins, so the abundance of about 1,500 proteins, and comparing them. And what you see really nicely is that when you grow on different substrates, the proteome groups really nicely, but you see that you have very distinct and very different proteomes depending on which substrate they're using. And for me, that seems really cool, because this is just one microbe. This is just an isolate that we have in the lab. And depending on what options it has and what kind of substrates it has available to it, it can basically entirely restructure its whole metabolism in order to do something else, which is a great trait if you're a microbe that lives in environments that are perhaps not that abundant or are that really changeable. Another really interesting thing that they do is that they can also use solid sources of electrons. So there's some nice work from James Byrne showing that you can actually cycle, do iron cycling in solid minerals like magnetite. So the same strain, Rhodopseudomonas palustris, will take electrons directly from magnetite. And they can also just kind of bypass any other complications and just take electrons directly from poised electrodes, which is a really cool, has a lot of really cool biotechnological applications as well, that there's a whole other field of research that are looking into. But one reason that I find photoferrotrophs really interesting is because one of my kind of other interests is thinking about what the Earth might have been like early in its history. So very early on in Earth's history, when you didn't have very much oxygen in the atmosphere, the oceans were primarily anoxic and are thought to be really ferruginous. And if you're a photoferrotroph, and all you need to survive is sunlight, iron 2, and a little bit of CO2, then this is kind of your, this is like your ideal scenario, because there's not so many things around that can really compete with you. You're not tied to any, you're not stuck to a hydrothermal vent because you need two, you need an electron donor and an acceptor. You can just live quite happily in the photic zone and use any of the iron 2, which is really abundant in the environment at that time. And it's thought that these phototrophic iron oxidizers contributed, at least in some part, to the deposition of some of these banded iron formations, which are really vast layered iron deposits that we find that were deposited out of the oceans in the Archean up to about 2.4 billion years ago, when we had the great oxidation event, and then the atmosphere was primarily then quite oxygenated. But when we think about how photoferrotrophs would have then evolved throughout the history of time, because the thought is that they are quite old, that they were around very early on in Earth's history. So the strains that we have today that we can now put in the lab, they've come from a whole history of different species that have probably had to adapt to massive changes in the Earth system, because the expansion, the proliferation of cyanobacteria would have meant that you have more oxygen in the atmosphere, and that they then start to outcompete photoferrotrophs in the photic zone. So they have the advantage that they can just live quite happily in the surface ocean, which is quite oxic, and then they'll gradually push down any of these strains which are obligate anaerobes, which don't like to have any oxygen exposure. So what would have happened over time is that as oxygen started to expand, these photoferrotrophs get pushed down in the water column and eventually kind of pushed out of the whole system. We find them today in places that are more like stratified lakes. So these are a few examples of places where there's been direct observation of anoxygenic phototrophs that are thought to do iron oxidation. So there's places like Lake Pavin in France, or Lake Metano, Lake Kivu, or Lake La Cruz. And these are all stratified lakes. So they're oxic in the surface, they're anoxic at depth, and then they have their redox transition is in the photic zone. And when you cross that boundary into the anoxic portion, if you look at the microbial communities in these lakes, you have a big bloom of anoxygenic phototrophs that really thrive under these conditions. And this is one of the reasons that we think that they were probably important and really thrived early in the Archean, because these are probably the closest modern analogues that we have for what the very early Archean ocean might have actually been like. And these microbes are they're pretty happy here. One thing we have started to think about more recently, when we're thinking about how photoferrotrophs might have changed over time, is that in the transition from an oxic to an anoxic, and from an anoxic to an oxic world, there's much more changes that happen to global biogeochemical cycling than just that now we have oxygen. So if you start introducing a lot of oxygen into the Earth system, you also oxidize lots of other biogeochemical cycles. And a key one that would be affected would be the nitrogen cycle. So as you have more oxygen around, you can have a much more oxidized nitrogen cycle than you would have had early on. And nitrogen cycle is really complicated even today, which is one of the main reasons that I actually like this diagram. So you don't have to understand everything about it. It's mostly to demonstrate it's really complicated. It has lots of intermediates. It's not a nice circle like the iron cycle. It's actually a bit of a nightmare. And it's, in some ways, almost impossible to reconstruct it in a modern environment, never mind in an ocean, more than 2.4 billion years ago. But what we can kind of confidently say is that if you don't have any oxygen around at all, your nitrogen cycle is dominated by reduced nitrogen species, things like ammonium, organic nitrogen. It's the transformations are kind of going between nitrogen fixation and ammonification of organic nitrogen. But once you have more oxygen, then you have a rise in this more oxidized side of the nitrogen cycle. We don't really know when this happened, but there's pretty good evidence that there would have been both denitrification and nitrification by about 2.7 billion years ago. So that's before we have the great oxidation event and the really widespread proliferation of oxygen in the Earth system. And the reason that's important, and the reason that I'm telling you about why we also need to think about the nitrogen cycle, is that we also have this other group of anaerobic iron oxidizers. And we've not been thinking about them very much in the context of early oceans. But I've been starting to think, along with a lot of my colleagues, that there would have been a role for these type of anaerobic iron oxidizers in an ocean which is stratified but not completely oxic. So the nitrate reducing iron oxidizers, they're a whole different thing. They get their energy from denitrification. So by transforming nitrate all the way into dinitrogen. And in the process, they either directly or indirectly oxidize iron 2. And like I said before, there's a lot of intermediates in the nitrogen cycle, and denitrification is no exception. So we also have a bunch of steps that we go through to go from nitrate to dinitrogen. So it goes through nitrite, nitric oxide, nitrous oxide, and then also dinitrogen. And there's two types. So one type of nitrate-dependent iron oxidizers will do this process directly. So they have the enzymatic machinery to directly couple iron oxidation to nitrate reduction. It's an autotrophic process, and they gain energy from doing that and fix CO2. But there's another type of non-enzymatic iron oxidizers. And what these do is they actually use carbon, organic carbon, as an electron donor, and they do nitrate reduction. So they are totally standard denitrifiers. But denitrification is pretty much in all cases a bit leaky, and it kind of leaks out intermediates in the transition from nitrate to dinitrogen. And a lot of these intermediates are very reactive with iron. So if, for example, you turn nitrate into nitrite, nitrite reacts really readily with iron 2 to form things like nitric oxide or N2O. And that's actually something that will happen with any denitrifier. So even something that's not kind of billed as an iron nitrate-dependent iron oxidizer, if you put any denitrifier in an iron-rich solution, then they will be able to oxidize a certain amount of iron. And this is something that I think about a lot, and the terminology is actually a bit complicated. And together with Andreas Kapler and a lot of our colleagues in Tübingen, we actually wrote a review a couple of years ago about this process and tried to clarify some things about the terminology, which is quite complicated. It's maybe a bit of a niche interest. I won't go into too much here, but there's a whole thing underpinning the way that we understand nitrate-dependent iron oxidation. But one thing that's important for this discussion and for the point that I ultimately want to get to with this is that in modern environments, we do see a coexistence of the two types of anaerobic iron oxidizers. So we find phototrophs and nitrate-reducing iron oxidizers together in modern systems. So for example, we see this in lake sediments and marine sediments where you have zones of where you have still a little bit of light in the top centimeter or so, and you still have nitrate, and you have iron 2. And we do find both types together in the same place. And that's the same in stratified lakes over a larger scale. So over a meter scale rather than a centimeter scale, we also find zones where these geochemical gradients overlap and where the potential for both photoferrotrophy and nitrate-reducing iron oxidation is there. So that takes me to the last part of my talk, to a question that I started thinking about kind of early in my postdoc and managed to continue pretty much since then, and hopefully we'll be wrapping up soon. And that is the kind of obvious question that all of these points and all of these considerations come to, which is, if you have these two types of metabolisms in the same place, if the geochemical gradients that they like overlap, do they compete with each other? Who would win in a scenario where they're both competing for electron donor, they're both competing for iron 2? So this is a lot of work from one of my PhD students, Verena Nicolai. I started it as a postdoc, and then when I got my junior group leader position, I was really lucky. I was able to hire Verena as a student assistant, and she just is one of the most talented people at cultivation that you've ever met in your life. So this is a lot down to all of her work. But what we were doing is trying to set up a really simple system where we could study the competition dynamics between photoferrotrophs and nitrate-dependent iron oxidizers. And the first system we decided to set up was to see what happens if we just put them both together in a serum vial and give them everything that they like. So we picked one purple non-sulfur bacteria, which is Rhodobacter ferrooxidans SW2, is a very common model strain for this kind of work. And then we picked an enzymatic nitrate reducer. So that was an autotrophic enrichment culture KS. This is the only kind of proven culture that we have that does iron oxidation autotrophically. So we didn't have many options on that side of the scale experimentally. So this is the one that we used. So we put them in serum vials, either individually or in a mixed culture. We give them loads of iron 2, so 10 millimolar iron 2. We give them 4 millimolar nitrate, which is enough for the nitrate reducers to oxidize 20 millimolar iron 2. So everything's in a huge amount of excess. And then we wanted to see how they would compete. So if we look at the iron oxidation over time, you have the nitrate reducer in black, the mixed culture in blue, and the phototroph in red. And what really stands out is that the mixed culture behaves basically identically to the nitrate reducer culture. It's so much faster off the mark to do iron oxidation that by the time the photoferrotroph even starts doing something, the iron is already gone in the mixed culture and it's completely used up. So what we can see is that if we have an excess of nitrate, then the photoferrotrophs will just be kinetically outcompeted because they're just not as fast. So then we thought, okay, that tells us what happens in one situation, but we're very rarely in a system which has so much nitrate. You know, what happens if you have a system where your nitrate is limiting, where you don't have enough nitrate around to oxidize all the iron that you have? Can you have a kind of coexistence? So we repeated this experiment, and instead of using 4 millimolar nitrate, we used 1 millimolar. And that means that it's only capable, if we look at the stoichiometry, of oxidizing 5 millimolar nitrate concentrations. So we would expect that once the nitrate reducer is done, there's some iron left over. And then we saw something that was really unexpected. And this is what kind of led us down this multi-year saga looking into how this process works. So what we saw is entirely contrary to what we were expecting, that again, the mixed culture behaves exactly the same as the nitrate reducer culture. So you can see here in black is the nitrate reducer, and here in blue is the mixed culture. And we get to this point where the phototrophs start to oxidize iron when they're on their own, and they ultimately use up all of the iron too. But in the mixed culture, the phototrophs never do anything at all. We never see any more iron oxidation than we would expect from the amount of nitrate that we have available for the nitrate reducer. So they're just not doing anything. And you see that reflected in the cell growth. So they're completely inhibited in being able to grow. Our cell numbers in the mixed culture max out at the same point as the nitrate reducer does. So this was a result that we weren't really expecting. that you have this inhibition, a complete inhibition of phototrophs in the presence of the nitrate reducing culture. And we did a whole bunch of test experiments that I don't have time to go into in a huge amount of detail here, because it was tons. We were doing it for ages. But what we were able to see from doing a lot of test experiments was that in order to see this inhibition of photoferrotrophy, we have to have cells which are alive and active. So if we add nitrate reducing inoculum, which we pre-killed, for example, by autoclaving, we don't see the same inhibition effect. And if we add living nitrate reducer cells, but we don't give them any nitrate, then we also don't see any inhibition. So what that tells us is that this inhibition of photoferrotrophy is just caused by something which is produced during active nitrate reduction. The question then is what? What causes this? Why is this happening? And if we go back to think about how denitrification works, you'll remember that I mentioned that there's multiple steps in this process. It's not that you go straight from nitrate to dinitrogen, you have steps in between. And conveniently for our detective story, two of those intermediates happen to be quite toxic. So nitric oxide and nitrous oxide are both known to be toxic to phototrophic bacteria. And lo and behold, we actually do see some production of both nitric oxide and of nitrous oxide in this culture. So NO is produced in very small amounts. We were only able to measure that in the nitrate reducer, not because it wasn't present in the others, but just we had to do this in a different lab, so it wasn't possible for us to measure all of the cultures. But in the nitrate reducer culture, when it's on its own, it produces about 25 nanomoles of NO. And then in N2O, we actually see quite a big accumulation of N2O in these cultures. And then it doesn't actually decrease, so it actually accumulates and stays in the culture over time. So that led us to a pretty easy hypothesis to test. If our hypothesis is that we have inhibition of photoferrotrophs because of some gaseous toxin, if we flush out the toxin, we should be able to solve the problem. So that's what we did. We set up the same experiment again, where we have the mixed culture in two different groups. In one, we don't exchange the headspace, we allow the volatiles to accumulate. And in the second, we exchange the headspace every time we sample. So we just flush it with nitrogen and CO2 to flush out any of the volatiles. And this was a big relief to see this plot, which showed that it actually worked. It seems it's so rare that you make a hypothesis and then you're actually right. But it genuinely works. So if you flush the headspace out regularly, you get rid of the volatiles, you see more ion oxidation. So you can see here on the left, this is much more orange than the one that was not exchanged. We see a complete ion oxidation in this case and incomplete oxidation in this case. And when we look at the cell growth, we see a much higher number of cells in the mixed culture when we exchange the headspace compared to when we don't. So we can relieve this inhibition by getting rid of these toxins. The question is, which one of the two actually causes it? So the easier one to test was to see whether nitrous oxide is toxic to our model strain. And we found out pretty quickly and easily that in fact it's not. So we can grow this strain at concentrations of nitrous oxide that are way higher than what we measure experimentally. So we don't think, despite the fact that N2O is in a higher abundance, that this is actually causing this inhibition effect. But what we can explain it with is the production of NO. And we had to go about this in a bit of a roundabout way because NO is so toxic that we couldn't get permission to work with it in the lab. So we went down a modeling route. Luckily, we managed to get on board Adrian Malage from Tübingen, who's a really talented modeler in terms of doing sort of microbial modeling. And what he managed to do was to model our entire system to get something that predicted a lot of the geochemical results that we already had. And what we can see in his model is that if you include a term for nitric oxide toxicity, so if you limit iron oxidation and cell growth by the amount or the production of nitric oxide, then you can perfectly predict the measured values of iron oxidation and cell growth that we see in the mixed culture. So you can see here in this solid orange line, that's the model including nitric oxide toxicity, and it fits directly to the measured values. And the same with the values for the cell numbers. If you remove this toxicity term, the model over predicts how much iron oxidation you'll have and under predicts how much cell growth you'll have. So we're pretty confident that we have enough pieces of the puzzle to see that this nitric oxide toxicity is actually driving the inhibition that we see. The immediate thought that we had was maybe we've just chosen a particularly weak and vulnerable model strain. So we repeated this with a bunch of other photoferrotrophs, so 2 green sulfur bacteria and 1 other purple sulfur bacteria. And in fact, we see this in all of the strains that we tested. And we even looked into the genomes of all of these strains. And what we can see is that they actually all have nitric oxide reductases. So they have a genetic capability to defend themselves against toxic levels of nitric oxide. So the one Rhodobacter ferrooxidans SW2, that has NOR-B, which is from the classical kind of denitrification pathway, whereas the other 3 all have NOR-V, which is not linked to denitrification, but it's thought to be specifically a defense protein for dealing with nitric oxide. So they actually have the ability to defend themselves, but in the situation that we're seeing here, it's just not enough to overcome this toxicity. We were lucky enough to be able to start working with some of our colleagues here in Bristol, so with Patricia Sanchez-Baracaldo and Giorgio Bancini, and they were able to, because they are very good bioinformaticians, much better than me, they were able to pull a whole bunch of phototroph genomes off of GenBank. So this plot on the right represents thousands of genomes that collapse down into about 450 species. And from interrogating the genetic capabilities of all of those genomes, we can see that NO toxicity defenses are actually very common in phototrophs generally. So more than half of all the phototroph genomes that we looked at contain at least one NO reductase that would help in this situation. Kind of interestingly, something that we hadn't picked up on before, but in fact, all of the chlorobi and all of the cyanobacteria have NOR-V. So it's something that is completely ubiquitous amongst those groups of phototrophs. Whereas NOR-B is a bit more common in the purple bacteria, which actually makes sense because a lot more of the purple bacteria do have some of the strains do have a denitrification capability. And I guess it makes sense that some of them might have kept this if it was particularly useful. So what that kind of tells us, not only is that the strains that we used for all of our experiments are at least representative of photoferrotrophs or of anoxygenic phototrophs generally, but that NO stress seems to be prevalent enough in the environment that a huge amount of species have evolved the ability to deal with it. And not only have they evolved it, but they've actually kept it. So for a whole group of the chlorobi or the cyanobacteria to all have this ability, tells you that it's something that is probably pretty important. So to kind of wrap that up, if we go back to our original question, what we originally wanted to know is whether or not phototrophic and nitrate-dependent iron oxidizers compete with one another. And what we see is that the phototrophs are kinetically outcompeted when you have excess nitrate, that the activity of nitrate-reducing iron oxidizers produces a lot of reactive nitrogen species, and that these have an inhibitory effect on phototrophic iron oxidizers. And what's the ultimate consequence of that is that we see an inhibition of photoferrotrophs that we would never have predicted based on the geochemical conditions alone, which is something that we do a lot. We go to an environment and say, okay, this is what the geochemistry looks like. This is the type of metabolisms that we might expect. But we have these community interactions that are something that we hadn't really thought about before. And these are also dictating whether or not a certain metabolism can proceed or not. So what does that mean if we're thinking about the early oceans, which is how I started this entire conversation? What we can see is that if you have nitrate reduction coupled to iron oxidation, this is something which could very feasibly have happened prior to the great oxidation event. When you have a bit of oxygen around, the nitrogen cycle becomes a bit more oxidized, so you have more nitrate around, but you don't have you don't have oxygenation of the deeper oceans. You still have a lot of stratified environments. And in this scenario, you could potentially have a coexistence of both types of anaerobic iron oxidizers. But if we start to then incorporate this potential effect of all these reactive nitrogen species that are promoted by the fact that you have very ferruginous conditions, this adds a whole other selective pressure to the photoferrotrophs that we just hadn't really been thinking about before. So we think that there's the possibility that this kind of scenario could have at least shaped the evolution of early photoferrotrophs and maybe even contributed to decreases in the precipitation of, for example, banded iron formations. So to conclude all of that, what I hope I've managed to convince you is that microbial iron cycling is really important in the biogeochemical cycling in modern environments and also in ancient environments. So I think if you take nothing but this from this talk, it would be great when you go through your own systems and maybe you have some interesting observations that you can keep in mind. Maybe there's some iron cycling microbes in there that might be having a role. But there's tons that we still don't know about their physiology. So I briefly mentioned it before, but we have so few isolates of these relative to so many other different metabolisms, especially the photoferrotrophs or the autotrophic nitrate-dependent iron oxidizers. We really need more models so that we can understand more about what their physiology is and how diverse they are and what they actually do and how they behave. And we especially don't know that much about how they behave and how they change in kind of real world geochemical conditions or how they behave as part of communities. And these community effects can have some unexpected sort of consequences. We've actually continuing this in a new project, or fairly new. We actually got a bit of funding from the German Research Council to look at this in a project with James Byrne and Andreas Kapler. And we have two really great, Verena we managed to keep, which was awesome. And we hired a new postdoc, Marcus, who's also working on trying to understand the metabolic flexibility of these phototrophs and how they behave in complex systems. So with that, I just want to thank lots of people, especially Verena, who did a huge amount of work on the content of this talk and was responsible for all the really cute cartoons. And also Leah, who started this project with me right in the beginning. And then all of our co-authors on that study, and especially a thanks to all of the Tübingen Geomicrobiology group. I can see that they turned up in force today, so it's nice to see all of you. And especially to Andreas Kapler, who was really supportive of all this work when I was in Tübingen, and it continues to be now that I've moved to Bristol. And with that, there's nothing to say but Merry Christmas, and take any questions you've got.
Thanks, Casey. That was fantastic. It's really great to see all those culturing experiments, some quite tricky, and the evolution, the questions about evolution that you're exploring and how this work has evolved now that you've carried it to your lab at Bristol. So it's really fantastic stuff. Thanks very much. So we'll open it up to questions, and I'll look for your questions in the chat, but if I don't see you, or if I don't see your little blue hands in the participants window, please feel free just to shout out, to turn your microphone back on and pipe up. I've got first question off the rank here, hit the chat pretty early. It was about, the question is, Casey, do you think there might be any other metabolites produced by the nitrate reducers that could be inhibiting the photoferrotrophs?
That's a really good question. That was something we thought about really early on, actually. And one of the first things we did was actually to test whether we were transferring anything from the inoculum into the culture that was actually inhibiting it. So we did a test where we filtered the inoculum from the nitrate reducer before we mixed it with the photoferrotroph, and you don't actually see any inhibition. So we can't rule out that they don't produce anything else or any other metabolites in the process. But I think the fact that we don't see any inhibition if we get rid of all the gaseous products, and we don't see inhibition when we do that experiment, makes me think that there's nothing major that's happening. But it's the whole metabolome is a whole other question. So you can never really rule anything out, but I don't think so.
Let's see, another question about nitrate reducers and whether the nitrite might play a role in inhibition at any step.
Yes, that's another really interesting question. That's part of this other suite of control experiments that we did. So what we can see is that the nitrite isn't toxic in levels that, on its own, the nitrite isn't toxic. So the photoferrotrophs have quite a high tolerance for nitrite. But if we mix nitrite and iron together, so let me rephrase that, the nitrite's not very toxic if you grow the photoferrotrophs on a different substrate. But if you grow them on iron, it is toxic. And we think that that happens because it's not the nitrite itself which is toxic, but it's the abiotic reaction between nitrite and iron 2, which then produces nitric oxide. So there's a kind of indirect inhibition. But we did actually test that, and you do see inhibition by nitrite, but only if you have this kind of abiotic NO production. But it's a really good question. We did think about that.
I might try to slip one of my questions in, partly because I'm really curious, but also partly to stall while I try to process some of the questions that are hitting the chat here. I might ask you guys to try to keep your questions a little bit concise, because some of you are throwing in some really great questions, but they're quite detailed. Okay, so actually, Casey, I was wondering that PCA plot that you showed of the proteomes that are clustering in very different directions. I just wondered if from that, or from looking at the proteomes and generally, if you get any sense of how these photoferrotrophs evolved different metabolic capabilities, like whether that sort of started off more as hydrogen oxidizers and moved across to various heterotrophic lifestyles, or because it seems as though they're at least mixotrophic. Is that correct?
Mm-hmm. That's a really deep question, actually. It goes to like the heart of a lot of this. You can't tell that so much from the proteome, but we have been thinking a little bit about what the very early environments for photoferrotrophs might be like. And I've been talking to Patricia Sanchez-Baracaldo a lot about the sort of phylogenies of anoxygenic phototrophs. And it's quite hard to pin down what they started as, because it was probably not doing some kind of heterotrophic metabolism. It was probably with iron or sulfide or hydrogen, but pinning down which of those it was is actually quite hard. One thing that we see that's interesting, so the green sulfur bacteria or the chlorobi are probably the oldest group, and they tend to be less keen to use organics than the others. Whereas we think that the purple sulfur bacteria are actually not as old. And within the groups, so the groups that the purple sulfur bacteria tend to fall in tend to be a lot more diverse. You have a lot of kind of normal sort of soil bacteria and things that are in there that are not even phototrophs. And they tend to be more flexible than the green sulfur bacteria and the chlorobi. But it's a really open question, but something that we maybe have a few hints about how it happened, but we don't really know yet.
That's interesting because you showed a difference between the nitrous oxide reductase pathways between the purple and the greens as well.
Yeah, they look like they're quite different, and there's a lot more kind of versatility in the purple ones. So I think there's been a lot more, kind of horizontal gene transfer and things in those. Although that's going a little bit outside of my remit of things that I know about, but.
A related question, but kind of coming back to the nitrous oxide issue a bit. Someone's asked, are there other possible molecules in the early oceans that may have served as essentially proxies for the kind of toxic reactions that you're seeing with nitrous oxide now? Are there other molecules that might be similar enough to nitrous oxide that the detoxification mechanism might target those as well? And how early do you think, if you can speculate about the importance of nitrate and nitrous oxide, how early do you think that might have occurred in the early oceans?
So one thing that's quite interesting is that so even in the complete absence of denitrification, in reactions with lightning, so if you have a kind of lightning interacting with reduced nitrogen species, you can produce NO. So there's actually even a corner of the literature that argues that aerobic respiration evolved from nitric oxide, and that's a bit, I don't know what I think about that, to be honest, but I would definitely say that we expect that we have NO at least in very low concentrations pretty much for the entirety of Earth history. So right from the very beginning. But could there be something that looked like NO that would make them evolve it? I don't know enough about it.
That's essentially part of the question.
Yeah, I don't know is the answer, but maybe. It's a really good point. Thanks. Was that Ben?
That was. Yeah. There's a related question from Alex about early sources of reduced nitrogen, and I hope that sort of helped to address that question as well. But if not, please, anyone, if you feel like your question hasn't been addressed, maybe they could email you later on, Casey. That'd be great.
I noticed Chow had Chow's a former PhD student, so I can say, Chow, send me your email. I don't have it anymore, then I can answer you.
I think there are some questions about details there in the culturing steps and potential limiting nutrients and vitamins and such that might be important. Anyone else for a couple of last quick questions before we wrap up today? Yep, Lucien?
Congratulations for the talk, excellent. I was wondering, when you purged the incubation, comparing it to the control, did you have constant pressure? I mean, after you purged, did you input nitrogen? I was wondering if you monitored the pressure in the headspace in the control and in the purged treatment during incubation.
Yeah, we didn't monitor it, but what we made sure of was that we didn't have an overpressure. So what we would do every time we took the sample or we purged it was to make sure you basically put a needle in and then put a little tube into a bit of water, and then you make sure we bubble out any of the overpressure so that it's equilibrated with atmospheric pressure. So we made sure we did think about that, but we didn't monitor it. We just made sure that there wasn't any overpressure or wasn't a big difference.
And about the reactive nitrogen species, I have a recommendation. Maybe you know Professor Jeff Cole from Birmingham. Jeffrey Cole.
No, I don't actually.
A very big specialist in reactive nitrogen species and also in the genetics of this.
Oh, thanks. I'll look him up.
I can send you an email later.
Yeah, that'd be great. Thanks.
Well, apologies to anyone whose questions I didn't get a chance to relay, but hopefully you'll get a chance if you want to take them up by email with Casey later, please do. And I just want to thank our speaker again for delivering a great talk today. It was really fascinating. Thanks very much, Casey. It was fantastic. Appreciate it.
Thanks so much for having me. Have a nice Christmas.
Thanks for everyone for joining us again today.